Right, in this video we're going to talk about the development of the current model of membrane structure, the Singer-Nicholson model, from a previous model called the Davson-Danielli model. These are your objectives. So before the Singer-Nicholson model, there was a model called the Davson-Danielli model, named after scientists who developed it, and it was really a sandwich model of the cell membrane. It was already known that the membrane was made up of phospholipids and proteins, both, and that the phospholipids were in a bilayer, just like we think of them now. And early electron micrographs showed a train track appearance like this, where you had dark alternating with light, alternating with dark in layers, dark, light, dark. And it was thought that that represented protein, and then phospholipid, and then protein. As well, water adheres to cell membranes more than it was expected it would if it were to be in direct contact with phospholipids. In other words, if this was the bilayer, and there were water in direct contact with it at times, here and here, it was thought that the results you'd get from surface tension experiments would have been different than what it was. So this was thought to be wrong. Therefore, the model was that there was a bilayer of phospholipids, and then instead of the phospholipids being in direct contact with an aqueous solution on either side, like we think of them being in places in the Singer-Nicholson model, instead there were proteins that were covering this whole surface and this whole interior surface. And so these would be globular proteins that were coating the surface completely. Not like we think of peripheral proteins now, here and there, in the Singer-Nicholson model. In the Singer-Nicholson model, there might be a peripheral protein there and an integral protein there, for example, but completely coating, and that's what it was thought was being seen here and was giving this result in experiments. However, evidence started to pile up against that model. For example, freeze fracture experiments demonstrated proteins embedded in the phospholipid bilayer. So what does that mean? Let's take a look at the diagram on the next page. This is from Campbell Biology, and this one on the left is showing a freeze fracture experiment. A cell is frozen and then it's split, and it tends to split in between the two layers of the phospholipid bilayer because of the weaker bonds in there. And it could be seen on the electron micrograph that there were proteins embedded in the bilayer like this, and they're showing up here like the little dots. And so that the existence of those embedded proteins on freeze fracture experiments is not consistent with this kind of picture. So that's a first piece of data that doesn't fit. Also, it was found that membrane proteins themselves are amphipathic. Remember, amphipathic means it has hydrophilic and hydrophobic regions. And again, that's not consistent with this model, because if the proteins were only on the outside, you'd expect them to be only hydrophilic, not hydrophobic. A hydrophobic protein, or hydrophobic region of a protein, implies that it's in a hydrophobic environment, like the middle of the bilayer here. So that's a second piece of information that doesn't fit with the Davson-Danielli sandwich model. Also, membranes in cells with different functions have different proteins in them. So different parts of a cell, or different organelles, or different cells entirely, have different proteins embedded in them, and that doesn't seem to be consistent with this kind of model here, where you've just got a layer that's quite static. Also not consistent with a static layer of proteins are fluorescent labeling experiments. When you fluorescently label membrane proteins and then you fuse two cells together, you see that the proteins move around. So what does that look like? Well, here's again a diagram to show that, and this one again is from Campbell. If you take a mouse cell and a human cell and you use fluorescent markers to label the proteins on them, that's what you're seeing here in red on the mouse cell and blue on the human cell, and then you fuse them together and you wait a little bit, you'll see that the proteins are all interspersed. Now, that wouldn't happen if they didn't move around. You'd expect that you'd have one part of the cell with the human proteins, and you'd expect that the other part of the cell would still have the mouse proteins and that they wouldn't intersperse. But the fact that they move around, and they do so quite quickly, is evidence that they are moving, and therefore that they're not in this cake-like layer on the inside and the outside. So that's another piece of evidence. So now we have all of this evidence against the Davson-Danielli model, and therefore another model needs to be proposed. And following this, Singer and Nicholson proposed the fluid mosaic model that we know of, and that's what we, with modifications, think of today as our model of the cell membrane. All right, so what's the relevance of this story? It's interesting, but why do we think about it? Well, it's a good example of falsification and how science works. Let me explain. In math, you can prove things true. However, in science, strictly speaking, we don't and we can't, in fact, prove things true. What we do instead, the way science works, is we suggest explanations, which we call hypotheses, with an E here if it's plural and I if it's singular, and we make predictions based on those, and then we test those by experiment and observation. And really what we're looking for is evidence against the hypothesis, just like we saw all of those pieces of evidence against the sandwich model hypothesis of the cell membrane. And if credible evidence comes up that is incompatible with our hypothesis, then we conclude that the hypothesis has been refuted or it's been falsified. In other words, that's not a correct model. Falsified or refuted, meaning that can't be the model that accords with reality. However, if we keep doing the right tests and we're really looking hard and we never find falsifying evidence, then what we do is we provisionally accept the hypothesis, meaning we accept it for now, but realize that there could always be evidence that turns up that refutes it, that falsifies it, and we can be quite particular about what that kind of evidence would be. For example, if we ever found credible evidence in the fossil record that there were humans and dinosaurs living at the same time, then we would have to seriously reevaluate our understanding of evolution. This also means that any hypothesis that can't be proven false is unscientific. A nice example is called Bertrand Russell's teapot. Bertrand Russell was a mathematician and philosopher, and he said, if you make the claim that there is a teapot orbiting in space at some unspecified distance around the Earth, and it can't be seen by telescopes, then there's no way of actually proving that false, because you could always say, oh, it was somewhere else, or you weren't looking in the right spot, and therefore that's an unscientific hypothesis. Now, just because something is unscientific doesn't mean it's unimportant. There are many domains of life that science just can't answer questions about, but it is important to think about what science can answer and what it can't. Any hypothesis that can't be proven false is not the realm of science. Okay, that's an introduction to the Davson-Danielli model and how it was falsified.